topic today I am going to discuss is about the transport across the cell membrane, or we can in this topic today I am going to explain about the active transport. Active transport in the sense, so whatever the solutes are present in and out of the cell membrane, the transport process that utilizes energy and occurs against the gradient is the active transport. So whatever the solutes like sodium, the glucose, the potassium, uh, whatever the solutes that are transported across the semi-permeable membrane at the expense of energy and occurs against the gradient, utilizes energy and occurs against the gradient, that is called as an active transport. We have three common characteristics of active transport. One is the uphill transport, and it requires energy. It exhibits saturation kinetics. Uphill transport, it requires energy. The third one, it exhibits saturation kinetics. So we can broadly classify the active transport into the primary active transport and secondary active transport. The primary active transport and secondary active transport. So these are the broad differences between active transport and passive transport. Against electrochemical gradient, nature of transport, it is along the gradient is passive. Utilization of hydrolysis of ATP in active and no need of ATP in case of passive. Saturation kinetics is a feature where may not be present in the passive. So what is primary active transport? The primary active transport is a transport mechanism that directly utilizes metabolic energy for the transport process. This mechanism is operated by ion pumps. So there are so many ion pumps. The solutes transported against electrochemical gradient with the help of energy, which is derived from ATP. Hydrolyze ATP to ADP. As the pumps hydrolyze ATP, they are called ATPases. So these are the types of ATPases: the P type, the V type, the F type, etc. And these are the examples: sodium potassium ATPase, calcium ATPase, hydrogen potassium ATPase, which are located on the plasma membrane, the sarcoplasmic reticulum, etc. So what is primary active transport? ATPases, the commonly occurring ATPase is sodium potassium ATPase or sodium potassium pump occurs common ATPases as calcium ATPases and H plus ATPase. ATPases are classified into P type phosphorylation, V type vacuolar, F type energy coupling factor type, and ABC transporters. Sodium potassium ATPase is an antiport pump. Pumps potassium into the cell and sodium out of the cell against the concentration gradient. Antiport transport system is primarily responsible for maintaining high potassium and low sodium concentration. So here this sodium and potassium, they are commonly called as electrolytes. See, sodium is a very important electrolyte which is present in the extracellular fluid, and the potassium is a very important electrolyte which is present in the intracellular fluid. So there should be an equilibrium between the sodium and potassium, and that equilibrium should not be disturbed. So coming to the structure and its functional aspects of sodium potassium ATPase. It is a heterodimeric protein made up of two subunits, alpha and beta subunit. The cytoplasmic side, alpha subunit has ATPase activity and the binding site for 3-sodium ATP phosphate. Extracellular side, we have alpha subunit binding site for 2 potassium ions and OABIN. So this is the structure you can see here. Alpha subunit and beta subunit, and we have the 2K binding site. So this sodium potassium pump was discovered in 1957 by a Danish physiologist, Jens Christian Skov. This fellow is from Dutch. Christian Skov of Aarhus University, who received the Nobel Prize for his discovery in 1997, and which was the culmination of his 4 decades of research aimed at explaining the mechanism behind the vital motor of the cells. He received the Nobel Prize in chemistry together with Paul D. Boyle and John E. Walker for the discovery of sodium potassium ATPase. So he was born in 1918. So what are the functions of sodium potassium pump? There is a P-type ATPase carrier protein. It is phosphorylated during the process. The pumps the 3 sodium ions out of the cell and 2 potassium ions into the cell. Cytosolic ion concentration, maintenance of cell volume, and supports the protein synthesis, etc. It maintains the resting membrane potential. Very important resting membrane potential in nerve. Sodium potassium also maintains RMP, resting membrane potential, by maintaining the ion gradients across the cell membrane. Heterome hormone actions. Sodium and potassium pump mediates action by many hormones of the cell. The example that is a thyroxine, aldosterone, insulin. So phosphorylation and dephosphorylation of carrier proteins lead to transfer of ions across the cell membrane. The binding of 3 sodium and 1 ATP molecule to their respective sites are subunit activates enzyme ATPase. It is called phosphorylation. So phosphorylation of alpha subunit by ATP causes conformational change in the carrier protein that transfers 3 sodium inside and sodium inside to the outside of the cell. The potassium binds to the outer side of the alpha subunit, and this leads to the hydrolysis of aspartic acid phosphate bond dephosphorylation. You can see this is the mechanism of action of sodium potassium pump. ADP, hydrolysis of ATP to ADP, and one sodium out of the cell and sodium inside the cell. Many hormones, chemicals, drugs act by increasing sodium potassium pump activities such as thyroxine, insulin, aldosterone, G-actin, etc. Many hormones and chemicals act by decreasing sodium potassium pump like dopamine, digitalis, metabolic poisons like DNP, 2,4-dinitrophenol, hypoxia, hypothermia, inhibit sodium potassium pump. Hypoxia means decreased PO2. Decreased PO2. Decreased PO2, hypoxia and hypothermia, decreased body temperature. Then calcium ATPase present in the cell membrane, membranes of endoplasmic reticulum, sarcoplasmic reticulum in the muscle cell. So it is a P-type. The pump actively transports calcium out of the cell and therefore maintains higher concentration of calcium ECF compared to inside the cell. So the calcium pump present in the membrane sarcoplasmic plays a very important role in the muscle contraction, maintenance, cytosolic concentration of calcium. It helps in storage of calcium in the cell organelles. Hydrogen potassium ATPase present in stomach, present in kidney. The proton into gastric lumen exchange of potassium. This is the primary step of HCl secretion in stomach. In kidney it secretes H plus into the tubular fluid, reabsorbs the potassium. Hence, it plays important role in the acidification of urine. The H plus pump or the proton pump is again on the membranes of the lysosome, endoplasmic reticulum, and mitochondria. HATPase, proton ATPase in mitochondria located in the inner mitochondrial membrane called matrix, cristae matrix. It is a F-type ATPase in organelles. Its main function is to synthesize ATP in mitochondria by utilizing energy and stored in the proton gradient created by the respiratory chain. So digitalis, this is the drug which is used in the treatment of cardiac disorders. Digitalis, routinely prescribed in the management of heart failure, inhibits sodium potassium pump by binding to the external surface of the alpha subunit. So up to this, what we have discussed about the primary active transport and different sodium potassium ATPases. Now we will see about the secondary active transport. The transfer of one solute again is concentration by using the energy generated by gradient of another solute. Usually sodium is the driver solute of most of these mechanisms. So most of the mechanisms, the sodium is a drive. Energy created by sodium gradient is utilized for transport of other solutes. The sodium gradient is generated and maintained by sodium potassium pump. So the typical example of secondary active transport is reabsorption of glucose from kidney, tubule or intestine. So whenever you take the diet which is rich in carbohydrates, and that carbohydrates is converted to glucose, and that glucose will be reabsorbed from the intestine into the blood or from the kidney into the tubule. That is the best example for the secondary active transport. So that's about the active transport mechanism. And if you have any doubts or queries, please subscribe the channel, and then you can freely express your doubts in the form of a comment so that I can take the feedback. Thank you.